All right, good morning. I'm Mark Jeske, Agronomy Manager for Pioneer, and I'd like to welcome you to the first installment of Season 2 of our Pioneer Agronomy Forward Thinking Farming webinar. Now, for those of you that might be joining us for the first time, the purpose of this webinar is to hear cutting-edge crop management insights directly from the experts on a wide range of different crop management topics. And the complete collection of our 2020 webinars is available at pioneer.com. You just search Pioneer Forward Thinking Farming, that will take you to the page. And we have around a dozen webinars there covering topics such as foliar fungicides, tissue testing, climate change, and getting the most from UAV and satellite imagery. For our webinar this morning, it's my pleasure to introduce Dr. Steve Strachan. Dr. Strachan brings to the table an amazing depth of knowledge and range of experience in the ag industry. His educational background is in soil chemistry and plant physiology, and he's held a number of different research roles in the Heritage, Pioneer, and DuPont organizations, ranging from seed corn production to herbicide discovery. And I've had the opportunity to collaborate with Steve on a number of different projects over the past several years, and can say without question that if you want to understand how and why corn plants do what they do, Steve is your guy. So this morning, Steve will discuss a bit about how corn plants take up nutrients and what that means for fertility management. So welcome, Steve.
Well, thank you. Thank you. Thank you, Mark. Well, good morning, everybody. We are ready to go into the slides, and we'll start the seminar. Okay. Mark, do you have the slides come up, or do I put them on mine and share screen?
You need to share screen.
Okay. So PowerPoint. Okay. Can you, did I share screen? Did I share screen yet?
Not seeing them yet.
Okay. Share screen. Share. Okay. All right. Now do you see the slide? First slide?
Yep.
Okay. All right. We are ready to roll. Well, this morning we're going to talk about feeding your corn, nutrition ratios for corn production. And with that, we'll go ahead and get started. There are 16 nutrients essential for corn growth. 2 of those nutrients, carbon and oxygen, the corn plant extracts from the air, just from the atmosphere. A third nutrient, water, or hydrogen, is extracted from water. What the corn plant does is it takes the water molecule, splits it into hydrogen and oxygen, and it uses the hydrogen to make sugars and cell walls and other organic compounds that it needs in the corn plant. And the oxygen is either consumed in the mitochondrial activity of the corn, or it's released into the atmosphere. And there are 13 nutrients that you have to be concerned about as a corn producer. The first 3 nutrients are called the macronutrients: nitrogen, phosphorus, and potassium. And they're macronutrients because they require hundreds of pounds of material per acre to meet those corn yields that are out there today. The secondary macronutrients are sulfur, calcium, and magnesium. And they're called the secondary macronutrients because they require about tens of pounds of material to meet the corn yields for today. And the third set of nutrients are the micronutrients. They are boron, chlorine, copper, iron, manganese, molybdenum, and zinc. And these are called the micronutrients because they are consumed by the corn plant anywhere from a few ounces to just a very low number of pounds per acre to support the corn yields of today. Okay, we're ready for the first poll question, Mark.
All right, we'll kick off our first audience participation question of the morning. You should see that pop up. First question: my current soil fertility program consists of nothing. I mine the soil because I rent the ground. I don't know if we'll get anybody admit to that. Standard rate of NP and K. fertilizing according to recommendations based on percent base saturation of CEC sites, and based on replacing nutrients removed plus adding additional amounts of selected nutrients. All right, it looks like we've got our results in on that. So far and away, leading answer is number 4, fertilizing according to recommendations based on replacing nutrients removed, plus adding additional amount of selected nutrients, with 72% of the vote.
That's really good. That's really good. All right. Thanks. Yes. All right. Okay. Well, and we're going to build on those answers and follow through on nutritional update and uptake in corn. Okay. All right. We'll continue on with the seminar a little bit. Corn is an amazing plant. In that corn plant, there's 89% of the total corn mass in the grain and stover consists of carbon and oxygen. And in that corn yield, if we have an average of 300 bushel per acre yield, over 3 tons of carbon and over 3 tons of oxygen are removed in the grain, and 900 pounds of hydrogen are removed in the grain. So if you think about it, that corn plant at 300 bushel per acre removes 12 tons of material that was created out of thin air, creates something from thin air. And in that process of making those 300 bushels per acre, that corn plant, that grain, has reserved about 25.9 million calories of energy. And it's an amazing plant to supply the food, feed, fiber, and fuel, and industrial products that make America grow. All right, we're going to look a little bit at the micronutrients now, or the other nutrients that you deal with in the soil. They're listed in the first column there, the various nutrients. And the second column is the percent pounds of nutrients per bushel of corn grain. So for instance, for every bushel of corn grain will contain about 0.6 pounds of nitrogen, or 0.42 pounds of phosphorus, or 0.27 pounds of potassium. And that ratio, that pounds of nutrient per bushel, remains pretty constant whether the corn plant yields 80 bushels per acre or 400 bushels per acre. That's pretty much constant across all of those yield ranges. If we look at a 300 bushel per acre corn yield, we're going to remove those numbers of pounds of nutrients in column 3. And then the fourth column is the pounds of nutrients relative to 1 pound of copper. And this is where we're going to start getting into the nutrient ratios. So for instance, if we look at the bottom, copper, 1 pound. For every pound of copper there is in the grain, there's 4,100 pounds of nitrogen in that grain. Or there's 337 pounds of sulfur in that grain. And from those numbers, we can calculate the various nutrient ratios in the grain. So some of the key nutrient ratios are, for instance, there's roughly about 12 pounds of N for every pound of sulfur. Or there's roughly 2.25 pounds of N for every pound of potassium. Or there's roughly 5 pounds of magnesium for every pound of calcium. Or there's roughly 340 pounds of phosphorus for every pound of zinc. So from that, we can see where the relative nutrient ratios come from in grain production. Okay, we're ready for the next poll question.
All right, second question. When I do fertilize, I'm concerned about primary macronutrients and soil pH, all macronutrients and soil pH, or all 13 soil applied and pH. All right, a little more even split on this one. So top answer at 45% is all the macronutrients and soil pH. 33% said all 13 in soil pH, and 23% primary macros and soil pH.
All right, thank you. It's those bottom two answers are the places where we really need to focus, and we're going to do that in the rest of the talk. Okay, so corn producer responsibility. What is your job as a corn grower? Your primary goal is to design a fertility program to supply adequate amounts of all of the nutrients, because whichever nutrient is limiting is probably the nutrient that is limiting yield. And a good place to start with all of these nutrient ratios is to look at the nutrient ratios of in the year. And we have this, published nutrient ratio guidelines based on those numbers. And something I hope to explain to you a little bit is we can rely on the corn plant, because the corn plant regulates nutrient uptake ratios to maximize kernel and ear growth. And what about nutrient ratios is important is that it's a very complex topic. So you're going to hear all kinds of discussions on nutrient ratios. And it's complex because we don't really know all of the answers to nutrient ratios yet. And what we know is that ideal nutrient ratios in the corn plant change throughout the growing season, and the rate of uptake of all of the different nutrients also change during the growing season. So these nutrient ratios within the corn plant is a moving target throughout the growing season. So what we're going to do today is we're going to talk about nutrient uptake in corn, and we're going to talk about what the corn plant does to regulate nutrients and nutrient uptake. So we're going to talk about total nutrients and nutrient ratios, the total amounts of those nutrients that are taken up and how they're taken up during the growing season, and nutrients removed in the grain. So this talk is going to be more of a plant physiology talk, and to help you explain what the corn plant does and why it does it. And once you understand that a little bit, you'll be able to think a little bit more about the importance of your nutrient ratios and how to work with the corn plant to get those maximum yields. Okay, so before we start talking about corn physiology, we need to talk about corn anatomy a little bit. We're going to start here. This is a cross section of a root, corn root on the left diagram. And the outermost skin of the corn root is called the epidermis. That's a layer of cells that's in direct contact with the soil. Just inside that epidermis, represented by the green, is called the cortex. And that's several layers of cells thick. And what that cortex does, it's like a holding place or like a gas tank for nutrients. The nutrients move from the soil, and as they move from the soil into the corn root, they move into the non-living portion of those cells in the cortex. Then the next layer, represented by the black ring, is called the endodermis, and that contains the Casparian strip. And that is the brains of the operation right there. What happens at the endodermis and the Casparian strip. And we're going to talk about that a little bit later when we get into the physiology. Now inside of that, the very central part of the corn root is called the stele. And what's important there is that's where the xylem and the phloem are. And the xylem runs nutrients and water from the roots toward the shoots. The phloem runs sugars and other nutrients from the leaves to wherever these nutrients need to go, whether it's roots or grain, or tassel, or wherever. And so the xylem and phloem are like a two-lane highway. One lane goes one way, one lane goes the other, and they keep interacting back and forth. They're always matched side by side. So the goal for the corn root is to get the nutrients from the soil, through the epidermis, through the cortex, through the Casparian strip, And that, as I said, that's the brains of the operation, to the xylem, where those nutrients are transported to wherever they need to go in the corn plant. Okay, that's a little bit of anatomy. Now let's figure out, let's talk a little bit about what that Casparian strip. If you look at the diagram on the right, you'll see the Casparian strip and the plasma membrane are represented with the black line, and they serve the same function as a rubber inner tube, rubber tire inner tube. And let me go through this. If we look at the left and look at the picture of the rubber tire inner tube, you've got the rubber membrane that holds the air inside the inner tube. You've got the valve stem that allows the air from outside the inner tube to enter into the inner tube. And you've got the valve core that holds the air in the inner tube. And that inner tube is impermeable so that the air outside the inner tube is separated from the air inside the inner tube. And basically, the Casparian strip and the plasma membrane serve the same function as that inner tube. They keep water and nutrients outside of the plasma membrane separated from water and nutrients inside the plasma membrane. So the area represented in blue in the diagram, that's the area that contains the xylem and the phloem for where we have to get the nutrients to. The area represented in green are those, is that holding area for the nutrients in the corn root. So, all right, we have the membrane covered, the Casparian strip and plasma membrane. Inside, in that plasma membrane are little holes represented by the white rectangles, and those are called plasmodesmata, and they serve the same function as the valve stem. They allow transport across that membrane. Now, inside of those plasmodesmata, those holes, are regulatory proteins, and that's represented by the brown rectangle. Those regulatory proteins in the plasmodesmata serve the same function as the valve core in the valve stem for the inner tube. So those regulatory proteins are the decision makers to allow nutrients to cross from one side of the plasma membrane to the other side of the plasma membrane. Okay, so those regulatory proteins are two types of proteins. They're called HATs and LATs. The HATs proteins are high affinity transport system proteins, and they extract nutrients from the soil when the nutrient concentrations are high in the soil. And they provide sufficient nutrient to enter the corn plant. Oh no, they supply sufficient nutrient to keep the plant functioning. So I made a mistake there. HATs proteins work when only there's very low concentration of nutrient in the soil. And we'll go through that a little bit later. So what they'll do is if your soil is testing very low in, say, sulfur, the HATs proteins will be effective, and they'll extract what sulfur they can to move it into the plant. And they provide enough, say, sulfur to keep the plant alive, but it still may show sulfur deficiency. So it cannot meet those high yields. For those of you who watch old Westerns, and the Indians are attacking the fort, and you've got this small group of people that are holding the fort down from protection of the Indians. That's what these HATs proteins do. They hold the fort down until the cavalry arrive. The LATs proteins are called a low affinity transport system proteins. And what they do is they're very effective when nutrient concentrations are present in high concentrations in the soil. And they're the real workhorse proteins. They move a lot of nutrient across that membrane into the xylem for transport. And they can supply sufficient nutrient to support those high grain yields that the corn plant delivers. So those proteins are basically the cavalry that come and save the fort during the battle. Okay, I'll hit you with some new terms here. Regulatory proteins, plasma membrane, plasma lemma, Casparian strip. These are terms you probably don't use much in your daily lives for your business. So what I'm going to do is I'm going to show you just in general how these proteins work. So first of all, and we have the green, those are the cells that are outside the Casparian strip, represented by the black. And the different rectangles are the different regulatory proteins. Each color represents a different nutrient. And what we have to do is get those nutrients from the right side of the plasma membrane to the left side for uptake by the xylem and the phloem. So there's nutrients in the outer part of the corn root. They're sitting there. And then what happens is those regulatory proteins move that nutrient across the plasma membrane. Each of the different colored circles may represent a different nutrient. So for instance, the light tan may be nitrogen, and the dark brown may be sulfur, and the red may be calcium. So there's a specific set of proteins that regulate each of the different nutrients. So they start to move nutrients across the plasma membrane, and they keep moving nutrients across the plasma membrane until for instance, the bottom one, the red X, the X with the red rectangle, until enough nutrient concentration gets high enough that one of those nutrients bonds back to that protein. And that's called feedback inhibition. And that when that nutrient attaches to that protein, that's like an on-off switch in a light switch. If there is no nutrient attached to the proteins are pumping nutrients across the plasma membrane and everything's going well. If there's concentration gets high enough that one of those nutrients binds to the protein, that protein is turned off and no more nutrient is transported across the plasma membrane. That's where the regulation for how many nutrients get across and eventually the nutrient ratios. So what happens is when the concentration builds up high enough inside the center part of the corn roots, those nutrients move to the xylem for transport to wherever they need to go. That reduces the amount of nutrients that are present in the central part of the corn root, and then the process continues again. So it's this process of turning on those proteins and turning them off that regulates how many nutrients move from the soil into the corn plant. So that's it in general. We're going to go through this with the HATS proteins in particular, because this is a new concept for a lot of you, I think. And we'll just go through this a couple times so you can see it in action. So for the HATS protein, the high affinity transport system proteins, they work when the nutrient content in the soil and the plant is low. So they'll come along, very low concentration of a nutrient. They'll move that nutrient across the plasma membrane so that it's in the center part of the corn root. And what happens is when the concentration gets too high in the center part of the corn root, it binds to that HAT protein, turns it off, and no more nutrient moves across until this protein is transported away. So the end result for the HATS proteins is the corn plant continues to grow even when there's very low concentrations of fertility in the soil. But it cannot transport enough nutrient across the membrane to meet the nutrient demand of the corn plant, so the corn may still show some nutrient deficiency and potential yield loss. So this regulatory protein works when there's very low concentration of whatever nutrient it is that's running deficient in the soil. Okay, that's the HATS proteins. Now let's go to the LATS proteins. These are the real workhorse proteins. They do not work if the nutrient concentration is low in the soil. What they do is they start to work when we start to see a lot of nutrients in the soil. And then they'll start to pump that nutrient across the plasma membrane into the center part of the corn root and for uptake. And they will pump a lot of nutrients across that membrane for uptake until eventually they run into the same regular, back up one, went too fast. They will keep pumping nutrients across that membrane until one of those nutrients binds to the protein and turns it off. So we have the on-off switch again. So it's this switch on and off that regulates, the corn plant regulates how much of each nutrient it takes up. And based upon the regulation of each nutrient, the corn plant determines the nutrient ratios it needs for maximum yields. So let's pull this together for both sets, for a case where you have, say, a low sulfur fertility soil and a high nitrogen fertility soil at the same time. If there's low sulfur fertility, represented by the gold, the HATS proteins may be active. They work in low soil nutrient concentrations. And the LATS proteins may not be active. So the end result is we're going to see a corn plant with possible symptoms of sulfur deficiency and reduced yield in that low sulfur soil. But if that soil also has high nitrogen, the HATS proteins may be inactive because there's too much for them to work. They become saturated. But the LATs are working just beautifully, and they're pumping across all of the nitrogen that corn plant needs to maintain the high yield. So, okay. That is a whole lot of physiology. That is something you're probably not familiar with. What does this boil down to in 15 seconds? If I'm a farmer and I want to know what does this mean in 15 seconds, or I need to tell a farmer what does this mean in 15 seconds, basically the bottom line is the corn plant has the ability to regulate whatever nutrients it wants to take from the soil into that corn grain. The corn plant sets the final ratios of nutrients in that grain. So the HATs and the LATs determine that. And they work whether the corn is yielding 80 bushels per acre or 400 bushels per acre. They work across all of those different yield potentials. And what they also do is they work during the entire growing season because the ideal nutrient ratios change throughout the growing season. So that's for the physiology that I've covered. The key point is the corn plant knows how to regulate the nutrients that it takes up to determine that yield. What's your job as a corn producer? Well, if there's no nutrient in the soil, there's no nutrients for those regulatory proteins to take up. So that's the key for you, is to make sure that there's enough of each nutrient in the soil to support yield. And it's the total amount of each nutrient in the soil that's critical, not really the relative ratios. As long as the relative ratios are close to what's needed in the corn plant, the corn is happy. And it's the total amount that you have to supply. Okay, so let's go to the poll question.
All right, next poll question. Bit of review here. Regulatory proteins for nutrient uptake in corn roots determine total amount of each nutrient that enters the corn plant, determine the ratios of nutrients that are in the harvested grain, change the ratios of nutrients in the plant during the entire growing season, or all of the above. All right, looks like we got most of the answers in, and far and away, top result is all of the above, with 83%.
Okay. Okay. So, as I mentioned, yeah, it is all of those things. It determines the ratios, the amounts, and it changes during the growing season. We're going to talk about changing during the growing season now. As I mentioned, nutrient ratios is a really complex topic. A lot of different people have a lot of different thoughts on this. It gets even more complex when we look at nutrient ratios during the growing season, because the ideal ratios that the corn of nutrients that the corn plant takes up during the growing season change as the corn goes through different growth stages. And one of the best studies in the literature is some research work done at the University of Illinois by Bender and his associates, Fred Bello and his associates, where they looked at corn plants, high-yielding corn plants, and looked at nutrient uptake during the growing season. So what we have on the x-axis for these graphs and for the next several graphs to come along is we have the nutrients, carbon, nitrogen, phosphorus, potassium, magnesium, sulfur, zinc, boron, manganese, and copper on the x-axis. And then on the y-axis is the accumulation of those nutrients as a percent of the total. So from planting to harvest, 100% of the nutrients are taken up during that time. From planting to V6, which is what the first graph is, we can see that anywhere from about 5 to 10, maybe 12% of all of the nutrients taken up by the corn plant are taken up at that growth stage. Now, if the nutrient ratios were constant across the entire growing season, then what should happen is that the size of the bars and the relative positions of the bars should move uniformly during the different stages of the corn plant during the growing season. And we see that from about planting to V6, that the size and the relative stages of the bars are consistent for that early growth of the corn plant. And that kind of makes sense. Corn's relying on the nutritional reserve in the seed to get started, and there's not a lot of growth between planting and V6. But when we go from V6 to V10 in the second graph, things really start to separate out. Between V6 and V10, about 20% of the nitrogen that the corn plant takes up is taken up during that growth phase. Almost 20% of potassium is also taken up at that stage, whereas maybe only 10% of the sulfur is taken up, or zinc is taken up at that stage. So the higher amounts of nitrogen and potassium that go into the corn plant at this time changes the ratios of nitrogen and potassium that need to be in the corn plant to support maximum yield. So those ratios change as we go from planting to V6 and then from V6 to V10. And the ratios continue to change as we move on to the different growth stages. If we look at from V10 to V14, this is when the corn plant is taking up the maximum amount of nutrients while the corn is in the vegetative growth stage. Almost 30% of the magnesium and boron is taken up at this time, for example. You can see that anywhere from about 20 to 30% of the entire nutrients are taken up during those 4 V stages, V10 to V14. By the time we get to VT, tassel, 70% of the nitrogen, potassium, and magnesium are taken up by the corn plant. And well over 60% of the boron and the manganese are taken up by the corn plant at this time. Whereas only 50% of the phosphorus, the sulfur, and zinc and copper are taken up by tassel time. I circled the carbon in blue, the first one, because this is kind of an interesting deal. When the corn plant is at the tassel stage, carbon is a measure of the dry weight of the leaves and stalks and everything above the ground plant for the plant. So only about 40% of the entire plant weight is present when that plant is at VT. So the remaining 60% of that corn plant is related to, for the weight of that corn plant, is related to ear production and grain production. So the corn devotes quite a bit of resources to ear and grain production in its life cycle. When we go from VT to R2, that's just pollination and early fertilization of the embryos. Not many nutrients are taken up at this time. And that kind of makes sense. The corn plant's gotten everything it needs in its vegetative stages. What it's thinking about now is the fertilization process and making sure that fertilization process goes properly. Now, when we go from R2 to R4, two things are starting to happen here. The bars in green are the nutrients that are taken up from the atmosphere or the soil. So if we look at nitrogen, basically there's maybe 5 to 10% of the total N is taken up from the soil to feed the ear at this time. Whereas boron, manganese, and copper, the corn plant's taking up all of those nutrients from the soil. Really high amounts of uptake of those nutrients at that stage. Now the gold bars represent the percent of the nutrients that are already in the corn plant, and those nutrients are being mobilized from the plant parts, such as the stalks and the leaves, and those nutrients are moving to the grain for kernel fill. So even though nitrogen, the corn's not taking up a lot of nitrogen from the soil from R2 to R4, it's taking about 10 to 15% of the nitrogen, total nitrogen, and moving it from the leaves to feed that grain. So a lot of the macronutrients in particular, nitrogen, phosphorus, potassium, magnesium, sulfur, and zinc, there's a lot of mobilization from leaf material to grain at this stage. Now, R2 to R4 is the stage in the reproductive phase where a majority of the nutrients are taken up during the reproductive phase of the corn plant. Now, let's go from R4 to R6, which is basically grain fill. This is where you get the kernel weights. And the first thing I want to point out is that basically 25% of the weight of the corn plant is added in those last few stages from R4 to R6. And that's kernel weight. That's where you get the big kernels between R4 and R6. Now what's interesting is if you look at a nutrient like phosphorus, during that latter part of grain fill, over 30% of the phosphorus and zinc are taken up from the soil to feed that grain at that time. And a lot of copper, high percent of the copper is going to feed the grain at that time. And then for the macronutrients in particular, you can see that that corn plant is moving a lot of nutrient material from the leaves and the stalk to the grain. So what's interesting about this is we get about 25% of the grain fill or dry matter weight accumulates during R4 to R6. And again, that's grain fill. And look at the nutrients that are removed in large quantities in the grain as that grain leaves the field. So for instance, 80% of the phosphorus that corn plant takes up leaves the field when that grain leaves the field. Or 60% of the nitrogen, roughly 60% of the nitrogen is in the grain. And that nitrogen leaves the field when that grain leaves the field. Or zinc, or sulfur. Some of those nutrients are really present in high quantities in the grain. And they leave the field when the grain leaves the field. So what's interesting about this is if you look at the nutrient contents in the grain, phosphorus, zinc, nitrogen, sulfur, and potassium, say, those are the nutrients that are most likely to show deficiency in long-term agricultural production of corn in that field. because those nutrients are being removed from the soil and leaving the field as that grain leaves the field. And what's interesting is it's not just the macronutrients, the phosphorus and nitrogen and potassium. We've got nutrients like, well, sulfur is a secondary macronutrient is important, but micronutrients like zinc and copper. So you may be starting to see some situations or hear of situations. Well, I'm starting to see zinc deficiency, and I've never seen it before. That may be going on because with these high grain yields, a lot of that zinc's leaving the field, and that zinc reserve in the soil is only so high. So, okay, let's go on to pulling this together now. So how do you manage nutrient ratios during the growing season? Well, the rates of uptake of each nutrient change as the corn growth stage changes. So the ideal nutrient ratios continuously change as that corn grows. And this is where you have to trust the corn. This is what the corn does. The corn has the ability with those HATs and LATs proteins to properly address all of those continuous changes so that it takes up all of the nutrients it can in the proper ratios that it needs to make the high grain yields. So what's your job as a corn producer? We're back to this again, because that's what's really important. What's your job as a corn producer? Your job is to make sure that that corn is happy and to make sure that there's enough of each nutrient in the soil to support each corn growth stage. Now, how you manage that job depends upon your soil type. If you're working with a lot of high CEC soils, there's a tremendous amount of nutrient reserve in those soils. So you may be able to fertilize only once or twice during the year, and there's enough reserve in that soil for that corn plant to take all of the nutrients it needs for the entire season. So those high CEC soils may be places like, Illinois or Iowa or those places like that. But if you're working with low CEC soils, say along, the eastern seaboard of Virginia or North Carolina, those little sandier soils, those lower CEC soils, you're going to have to manage nutrients in a different way because the reserve of those soils is low enough that you're going to kind of have to spoon feed those nutrients throughout the growing season to maximize the corn yield, because the soils do not have the ability to store a lot of those nutrients. So you have to feed those nutrients kind of as they're needed. So an ideal nutrient ratio in June might be a little bit different than the ideal nutrient ratio in July. And that's something you have to work with as a corn producer. So, okay, let's pull this all together. So in summary, we're going back to the beginning. How do you determine the correct nutrient ratios in your fertility program? So a good place to start is to start with the ratios of the nutrients harvested in your grain or your silage. We have nutrient ratios worked out for whole plant, corn plant for silage. So we've got similar numbers for silage as well. So you start with these ratios, and then you fertilize to replace those nutrients lost during harvest in the grain or in the silage. Plus, you add nutrients as your budget and business plans allow. And the purpose of adding some additional nutrient is to build a nutrient safety buffer zone in your soil. And once you know the total amounts of nutrients you want to have, consider the amounts of nutrients already in the soil, because those are already available for corn uptake. And then balance these different things out. Make sure that they're adequate amounts of all nutrients, not just N, P, and K, but all nutrients, because you don't want to deal with what's called a hidden yield loss. Some of these nutrients may be in concentrations low enough that they won't allow for maximum yield, but yet they're still high enough that you may not ever see a deficiency of these nutrients. So it's be careful to make sure there's adequate amounts of all nutrients, so there's no hidden yield loss. And then fertilize. Your fertilizer plan will be according to what your soil CEC capabilities allow, whether you can fertilize all at once or you have to spoon feed that fertilizer out through the season. That depends upon your soil reserve. And the key thing is make sure you have enough nutrients to add. They don't need to be exact ratios. They just kind of need to be close, because the regulatory proteins in the corn plant will establish those final nutrient ratios to get that maximum yield based upon whatever the soil environment allows. So, okay. Question.
All right, we'll go into our final polling question now. Your job as a corn producer is A, to supply replacement amounts and build up additional amounts of NPK and S and adjust soil pH for maximum yield. B, apply nutrients in specific ratios to replace nutrient amounts removed in the grain. Or C, apply replacement amounts of all nutrients, build up amounts of selected nutrients, and rely on corn regulatory proteins to establish ideal ratios. And we'll give that a minute for the votes to come in. All right. So we've got 94% with the third answer. Apply replacement amounts of all nutrients, build up amounts of selected nutrients, and rely on corn regulatory proteins to establish ideal ratios.
All right. Okay, thank you. Yeah, it's nutrition, corn nutrition. It's just absolutely important. The goal is to make that corn plant happy. If the corn plant's happy, the corn plant's going to yield. All right, I think we have time for a few questions, if we have some.
Yes, we do. And if you have questions you want to submit, you can use the Q&A button at the bottom of the window. We'll take a few of these before we close out our time here. So interesting question that came in, getting back to the function of the HATs and the LATs proteins. With the HATs proteins taking up nutrients at low concentrations, is the plant expending more energy for those proteins to work?
I think the answer is no. The plant is not expending more energy for the proteins to work. I like to describe the HATs as they're hunters. They're there, they can see something at very low concentration, and they can extract it from those outer cells very, very efficiently. The LATs, well, it's kind of like my wife and I. My wife can find things that I just can never see. But if it's there right there in front of me and there's a large amount of it, I will see it. And that's where the low affinity proteins work. They're really more gatherers. They see things right there, and they're the workhorse proteins that move a lot of those nutrients across the system there. But the energy required for the HATs or the LATs, corn plant, really no difference in terms of expenditure of energy for that. Now, where energy will come into play is if there's a low concentration of a nutrient in the soil, and the corn plant will try to compensate for that and spend more energy for root growth. And if it's spending more energy for root growth, that's less energy available for shoot growth. So that's where the energy expenditure comes in.
All right, thank you. Got a question about nutrient ratios in grain and how yield level affects that. So is the nutrient ratios in the grain the same at 300 bushel corn versus 100 bushel corn?
Pretty much so, yes. Let me see if I can back up. Right here, this slide right here, are you seeing the table on the screen?
Yes.
Yeah, okay. That second column, pounds per bushel. And several research papers have shown this, and a bunch of different studies have shown this. Those pounds per bushel, there's a range. There's a range of pounds per bushel for each of the nutrients, but that range is fairly consistent whether it's 100 bushel corn or 400 bushel corn. So that concentration stays the same. The total amount leaving the field changes, but the concentration stays pretty close.
All right. What about luxury consumption? That's something that we hear about with some nutrients where the plant is taking up more than it actually needs.
That's a good question. Luxury consumption to me is like, well, it's like you and I going to lunch. Sometimes we eat more calories than we need to eat because we have the opportunity to eat more calories than we need to eat. Luxury consumption is, I think what the plant does is it takes those extra nutrients and it stores them in either the vacuoles or somewhere it stores them. And it's kind of like having excess inventory. If for some reason, for instance, I take up extra potassium early because the season is wet or, good rain. And then in August, it gets a little bit dry. And that's an entirely different topic about how nutrients move from the soil through the soil water to the plant. If potassium uptake is a little bit low in August, it can say, all right, I've got these extra luxury consumption nutrients I stored earlier. We'll put those back into circulation and we'll use them then instead of, because we can't get them from the soil. So there is some luxury consumption. It's probably a good thing in the corn plant, yes. I'm not sure I fully answered that question properly.
Another question here. Have we seen a shift, as far as you're aware, in nutrient uptake or ratios with newer hybrids compared to older hybrids, say from the 70s or 80s?
I have not made that comparison directly looking through the literature. If there has been a shift, it's been a very small shift. Where the shift has occurred is the yields are getting so high now that the total amount of nutrient leaving the field is, we're really mining those soils harder now than we did, say, in 1950. So fertility, sustainability in agriculture is going to rely on more fertility because more of that fertility is leaving the field when the grain leaves the field.
Yeah, that's 2 points I'm aware of relative to that. As you said, just the total amount leaving the field is greater than it's ever been. And I have seen some studies that the timing has shifted a little bit, at least for nitrogen, that we see corn plants are continuing, newer hybrids take up more nitrogen post-tasseling than older ones.
Okay.
So it makes that the nitrogen supply later in the season is a little bit more critical than it was with newer hybrids than it was previously. I have one final question here that we'll hit before we close it out. Related to that, do we see much in the way of differences among hybrids in their ability to take up nutrients, or are they all fairly similar in that regard?
The hybrids are pretty similar. The University of Illinois study that I referenced here was done with, I think it was 5 or 6 different hybrids, and they checked for hybrid responses, and they did not see any differences in the hybrid responses. So they were able to pool all of the hybrid data together to generate the final responses that they reported.
All right. Well, we're getting close to time here, so I think with that, we'll close the Q&A. So thank you very much, Dr. Strachan, for your presentation here this morning. A couple housekeeping items here before we let everybody go for the morning. One, we were made aware of an issue during the webinar that apparently Zoom has an upper limit on the number of participants, and we bumped up against that limit today. So we had not everyone who registered was able to get in. So the recorded presentation will be posted to pioneer.com. So if you know anyone who wasn't able to connect live this morning, they'll be able to watch the presentation later. And then we have a full slate of additional webinars coming up through the winter and spring here. So be sure to join us for the next webinar, which is scheduled 2 weeks from today on February 5th, where we'll be getting a behind the scenes look in a soil lab to see how soil testing takes place. So with that, thank you, Steve, and thank you all for joining us here this morning for the Pioneer Agronomy Forward Thinking Farming webinar.
Thank you.